It's my great pleasure to introduce to you once again our recent inductee that we saw earlier, Professor Luis Marzan, who is currently Professor and Scientific Director of the CIC Biomagun, where leads the Laboratory of Nanoplasmonics. Professor Luis Marzan, as his research interest is on the synthesis and assembly of nanoparticles, nanoplasmonics, and the development of nanoparticle-based sensors and diagnostic tools. He has traveled quite a bit. He's been visiting scientist and/or professor at the University of Vigo, Utrecht, Tohoku University in Japan, University of Michigan, University of Melbourne in Australia, Hamburg, and then the Max Planck Institute. I'm happy to introduce then to you our medalist, Blaise Pascal medalist for material science, Professor Marzan, who, in recognition of his contributions to the understanding of metal nanocrystal growth and self-assembly, plasmonic properties, and sensing applications.
Good afternoon. Thank you for staying until the last session. Thank you to the European Academy of Sciences for admitting me as a member and subsequently awarding this very prestigious Blaise Pascal medal. I feel really honored to have been selected. I would also like to thank a couple of the previous speakers because they actually introduced quite a number of concepts that will be useful to follow my lecture. I will try to show you today a little bit of an overview of different parts of the research that we do in the group already for a number of years. I use the title Colloidal Nanoplasmonics because it summarizes rather faithfully the two main topics of the research that we do. So again, I start with Michael Faraday. This has not been pre-agreed with Professor Mingus, but it saves me some time to actually introduce the work by Faraday. In fact, the relationship between this paper and what we are doing today is actually very close. So indeed, Faraday was working with metals for some time. I think this pointer doesn't work very well, but anyway. So he was working with metals for some time. He was doing indeed the first reported colloid chemistry, which did not have a name. And he was actually trying to understand why, by playing with the dimensions of metals, he was seeing different colors and studying what happens when you gradually reduce the size of the metal. And in fact, he was playing with two types of materials. One was the dispersion of particles that was already mentioned, where the color of the dispersion depended on the way that he was doing the chemistry. And in this article, which is I always recommend to read, he actually describes and tries to propose ideas of why the colors are changing and what happens to the metal to induce that color change. So actually, there are, in that paper, there's not only the preparation of colloids, it also discusses, He also discusses effects of light scattering, the aggregation of colloids with the addition of electrolytes, quite a few concepts that were rediscovered much later and explained more properly when suitable characterization techniques were available. But Faraday was also working with thin metallic films, and he was somehow beating small metallic leaves and making them thinner and thinner, trying to observe if at a certain point, light, visible light, can actually go through the film. And in this picture here, it's not very clear, but if you look close enough, you'll see that there is the typical golden reflection, but also in the shadow, you can see some bluish tone, which is related to this dip in the absorption band of the film, meaning that light around 500 nanometers can actually penetrate partially and go through the film. So these are true size effects, which have nothing to do with the chemistry of the material. And actually, this is what I try to show in this picture here. If you take one of the particles from the bottles of Faraday and take it to an electron microscope, if you measure the lattice constant, you will find exactly the same lattice constant as you find in bulk gold. So the only reason why these colors are completely different, coming from exactly the same material, are the dimensions of the material. Okay, so we work on trying to improve what Faraday started 150 years ago. So we do a lot of, again, wet chemistry to grow metal particles. In the diagram that Professor Mingo showed before, we actually go to the other side. So we try to grow a bit larger particles, because I'll show you in a minute, only for larger particles, we see this very beautiful optical response that are related to electron oscillations. So we do it in a way that we try to engineer the process to actually generate not only particles that are hopefully all equal to each other, but also that we can engineer the morphology of the particles. And this is important because the actual optical response of these systems will depend very strongly on very minute changes in the shape of the particles. So once we know how to do that, we actually use theoretical models to understand this optical response. We modify the surface chemistry of the particles, and we try to exploit that to generate organized assemblies, either in 2D or in 3D. And then by understanding and applying the optical response of either the signal particles or these assemblies, we find applications related to biosensing and drug delivery. Okay, so this diagram shows you why we have these very special optical properties. And this is related to metals having free electrons. So because there are free electrons, when an electromagnetic radiation comes, you can actually generate surface waves within the metal. And so each metal will have a characteristic bulk plasma frequency, so the frequency of these waves within the metal when they are excited with light. But if you have a very thin metal, these waves do not fit inside that thin film anymore because the wavelength is too long. So actually these waves are confined at the interface between the metal and air, water, or whatever dielectric material is located next to it. And in fact, this is the basis of commercially available surface plasmon biosensors, because you can bind biological receptors to this metal film, and then you will see changes in the actual reflection of light when a biomolecule binds to these receptors, so you can identify selectively the presence of these molecules. Something similar happens when you reduce the dimensions to very tiny particles. And so if the particles are very small and spherical, the electric field of light will induce the formation of dipoles, separation of charges, and then the actual oscillation of the electric field in the radiation will couple with the oscillation of these charges, and then you will have a specific frequency at which there is this coupling. And then at that frequency, there will be a very high absorption of light that provides the beautiful colors to the dispersions of silver or gold particles. So these pictures here show you the yellow appearance of a silver colloid and the red appearance of a gold colloid. And excuse me that I insist that I'm not really being precise when I say the yellow-silver color or the red-gold color, because again, if you change the size of the particles, or especially the shape of the particles, the color will be different. So if one, I hope my take-home message today will be there is nothing like a golden color or a silver color, because both gold and silver can have any color, depending on the shape and the morphology of the particles. Another interesting phenomenon is that when you excite these charge oscillations, you actually create very high electric fields exactly at the surface of the particle. And this can have a very large effect on molecules that are binding to the surface. So I'll get back to that in a minute. But this is important for the applications. Now, if we try to understand and to predict which will be the wavelength or the frequency of the oscillation of the electrons for particles of different morphologies, in principle, we have to resolve Maxwell's equations completely. And unfortunately, we can only do that, or maybe fortunately for the mathematicians, we can only do that by using numerical methods. So what we usually do is we take the particles that we want to simulate, we divide them in very tiny pieces that can be treated as a dipole, and then we apply the equations to calculate the interaction of light with that dipole, and then we integrate over the full volume of the particle or the assembly of particles. So now this can be done very efficiently nowadays, so we are really lucky that we can understand almost precisely how a specific particle will respond to irradiation of light. And so, if it wants, oops, okay. And so now we are able to make particles of different morphologies, spheres, but also nanorods. And what I show you here in colors are experimental and theoretical determination of the enhanced electric fields on the particles when they are excited with the frequency that drives this electron oscillation. So you can see that we can calculate exactly the same behavior as we find experimentally, not only for spheres and rods, but also for bipyramids, for cubes, for nano dog bones, for nanostars, for triangles. So the zoo of metal particles that we have at our disposal today is really very varied. And one of the main objectives of the research in my group is trying to understand why, by changing the reaction conditions, we can actually drive the growth of the crystal into these very beautiful morphologies, and how then we can exploit that knowledge to devise new methods so that we can actually get to other morphologies that can be more beneficial for a specific application. So nowadays we can do all this, but we only understand a little bit of that. So now we can also predict how these particles can help us to find a use for them. And there are actually many potential fields where these properties can be useful. One of them is improving the efficiency of Raman scattering spectroscopy. So very briefly, I don't want to bother you with details, but in Raman scattering, what you have is, rather than exciting an electron to a higher energy state and then see how it comes back, you actually excite an electron to a forbidden state, and then you watch how this electron comes back to the same electronic state, but to a different vibrational state. And so with that, you're able to identify exactly which are the bonds present in the molecule. So this is a very specific spectroscopy that provides you with what we call a vibrational fingerprint of the molecule. The problem is that because you have these forbidden states involved in the process, the probability of this process is extremely low. We normally say that only 1 in 1,000 million photons is scattered in this way. So just imagine you need at least 1,000 million molecules to actually see a proper signal. And if you want to detect, for example, disease biomarkers at very low concentrations, so at very early stages of the disease, you're never going to manage with this technique. For some molecules, this can be improved, but actually the nice thing is that in 1974, in the group of Martin Fleischmann in Southampton, they discovered that if molecules are located on top of a rough metallic electrode, in this case silver, the signal that you obtain for the same number of molecules is many orders of magnitude higher. Incidentally, and well, just don't take it as a criticism, this is the same Fleischmann who discovered the cold fusion. But anyway, so in this case, the effect was real. Unfortunately, the explanation in the paper is not correct. So what they proposed is that because the silver electrode is very rough, you actually have many more molecules than you initially estimated, and this is why you get this very high signal. But you can really calculate the absorption density of molecules, of pyridine molecules on this electrode, and you will never come to those numbers. And so then there were immediately a few other people trying to explain. Andreas Otto proposed that there is electron transfer between the metal and the molecule, and indeed these effects exist, but again, the enhancement that you would get with this effect is not accounting for the experimental observation. So actually now we believe that what was proposed almost simultaneously in the groups of Van Dijn, Creighton, and Moskowitz is the main effect driving this enhancement. And this is related to this enhancement of electric field on the nanostructured metal. So this electric field actually affects the polarizability of the molecules and improves a lot the efficiency of the Raman scattering process. So now it's the moment that then we can apply what we know about growing particles, because if we can optimize the electric field, then we will improve the sensitivity of the technique. So what you need is to have the molecule adsorbed on the surface of the particle, then this represents the vibrational fingerprint, and then the electric field will take you to potentially enhancement factors up to 12 orders of magnitude, meaning that in theory, and I should say has already been demonstrated by several people, you can actually apply this technique to detect one single molecule. And this is extremely powerful. The problem is that if you want to do it correctly and quantitatively, you really need to understand very well the optical response of the material that is enhancing the signal. And so you can engineer the morphology of the particles, as I already said. And we know that particles that have sharp tips create much higher electric fields. But we also know that if particles come close to each other, they also generate very high electric fields at the gaps between them. So for example, in this spectrum here, what you see is a configuration like this with molecules adsorbing on spherical particles. When they are separated in a colloidal dispersion, the enhancement is not enough, so we don't see anything. This is the blue line. But if we add sodium chloride, this is the effect that already Faraday described in his paper, we generate the aggregation of the particles, so we generate many of these gaps where the electric field is very high, and then you immediately see this very well-defined spectrum that tells you exactly which molecule is present in the solution. Okay? So you can either play with the shape of the particles or you can play with the organization of the particles. And this is what we try to do. So now, in order to try to understand how these small crystals grow, you need to at least try to follow some of the classical theories telling you how particles can evolve in solution depending on the concentration of the precursors and how fast the reaction goes. And so what we normally try to do is we try to separate the processes of nucleation from growth so that we can actually somehow affect the growth on well-defined seeds. And also to avoid that seeds are formed continuously during the reaction, which would lead to this very disparate distribution of particle sizes. So once we know this, we can actually go and use something similar to what Professor Mingos was showing to make his very small clusters, using a very strong reducing agent to reduce gold chloride into tiny particles that then we use as seeds to grow the larger particles that we need. Interestingly, in the processes that we normally use, depending on other additives that are present in the solution, these very tiny seeds can either be perfect single crystals, or they can have crystalline defects like these planes. It's very difficult to see. But actually, there are some lines here and here that show you that this is not a single crystal. So there are several crystals that are packing to it and close to each other. And now, if we promote the growth of more gold on these particles by using a weaker reducing agent, we actually can obtain these elongated particles. And if we start from the seeds with defects, we obtain elongated particles that maintain this pentagonal cross-section because of the defects. If we start from the single crystals, we can also get elongated particles, but they are actually single crystals. And now we know that there are other molecules that are adsorbing on the surface, which are probably not only stabilizing the particles in solution, they are actually determining the shape into which the particles will grow. And then if you have access to the modern electron microscopes, you can also see the morphology of the particles in 3D and even the distribution of the atoms in 3D. And then you can really understand how these molecules can actually affect the distribution of the atoms and the formation of these elongated shapes. And so with this, we try to design changes in the chemical process so that we can obtain different morphologies. And so nowadays, we can prepare gold nanorods of this quality. They are almost monodispersed. In fact, we now know of ways to actually make them perfectly monodispersed, at least in terms of optics. And this will be published next week. But for now, it's enough to know that by simply changing the reaction parameters, we can actually tune the morphology in a way that the optical response is gradually shifted. So we can find exactly the morphology that will absorb light in the color that we want. And so, as I told you, there is no golden color. All these absorption spectra are for gold, and they are all different and leading to different colors, only by changing the ratio between the length and the width of these nanorods. Okay, now, going into organizing the particles, actually there are many reasons to do this, because not only when you bring particles close to each other, you generate electric fields in the gaps. In fact, coming back to Professor Balsani's presentation, this is something similar to what he presented for atoms, but now we are presenting it for particles. So we bring, for example, two nanorods close to each other, and we generate bonding and anti-bonding plasmon modes. So you're actually generating a new material with different properties. If you bring particles in a sort of aromatic fashion, you may have a completely different way to manipulate the electrons in this nanostructure. If you generate helical templates and then you attach your particles in a helix, you will obtain chiral plasmons, and this has been demonstrated. So we can think of many different ways to really drive the assembly of particles to obtain completely new properties that have not been demonstrated before. And this is what we are trying to do, and we are trying to exploit also long-range, perfectly organized assemblies of particles so that the electric field that we generate at the gaps is exactly the same everywhere and as high as possible, so that we can detect the more faithfully possible the presence of certain molecules in solution. So again, theory can help us not only to predict the properties of a single particle, but also the properties of collections of particles. And so here, for example, you see calculations that tell you that if you change the gap between the nanorods when they are placed next to each other, you can gradually shift the position of the band. This is not a big surprise. But actually, with the modern calculation methods, you can even compare the optical response of a completely random film of thousands of nanorods to the same number of nanorods, but located in a crystalline fashion. So forming multi-layers where all the nanorods are standing up. And so again, I'm not going to go into the details, but what we see in these pictures here is that the film is only efficient very close to the surface, while in the crystal, you obtain very high electric fields at every plane in between monolayers. So if we're able to fabricate this system, we will be in a very good business to really detect molecules from solution if we are able to take them in between the monolayers. And now we go again to the fabrication, and then we think we can probably induce the assembly of particles by using templates. And so we do a combination of what is called bottom-up and top-down approaches in nanotechnology. So we use the top-down to create holes in a PDMS template, and we use the bottom-up to fabricate the nanorods, and then we simply deposit a drop of the nanorod solution on a glass surface. We press the stamp with the holes of the sizes that we want, and we let the solvent evaporate. And the solvent in this case is water. And then what we hope is that the nanorods will get organized perfectly within the holes of the template. And so we do the process, and we compare what happens without the template and with the template. And as you can see here, the bright spots that indicate where the particles have actually been organized are completely random without the template and follow the morphology of the holes with the template. So this is already a good indication that at least we get organization within the holes, and we can pattern these assemblies. Now, after optimization of this process, you can go to the electron microscope, and then you can see a perfectly organized assembly of towers. And all of these towers are made of many thousands of nanorods in an organized fashion. And why do I say in an organized fashion? Because here we can actually, the most we can see the outer faces of these very big towers of nanorods. But again, by using modern electron microscopy, we can cut slices and observe every plane inside of these towers. And we can do reconstruction in 3D. So here you see everything inside of this so-called supercrystal. So we know that there is a very close, a very good organization close to the ideal crystalline lattice. And actually, you can, with these images, you can assign a directional vector to each of the particles, and then you can calculate a distribution of angles, and you actually see that it's very narrow. So this is close to the perfect system that we wanted to make. The problem is that in these assemblies, the planes between monolayers are very, very small. So it will be very difficult for molecules to get in between and fill the very high electric fields. So then we apply more chemistry, and what we do is to actually fill these supercrystals with a mesoporous oxide. And we are so lucky that we see that in the process, the monolayers actually get shifted a little bit from each other. And if we look carefully enough, we see that there is the silicon oxide in between. And again, in the 3D, you can see that the organization is close to perfect. And now the molecules can actually diffuse to the intermediate layers between the gold nanorods. And now, so now we go to the application. So this is a first evaluation of the enhancement of the Raman signal of model molecules. But I was lucky enough that the ERC also trusted me to carry out a very ambitious project. And actually what I showed you so far was also part of the project, because this was necessary to really accomplish the objective that we established at the beginning of the project, which was related to understanding how bacteria talk to each other. And this was something that was really very far away from my expertise at the time, but having the funding of the ERC was enough to recruit a microbiologist, discuss everything that we need to do so that we can try to understand these processes, and then apply our techniques and our materials to really carry out this important objective. So the idea is that when bacteria are floating around, they only act when there is a sufficient number of individuals in the community. It's like when I came back from the coffee break, only Professor Serpong and I were present in the room. So if this was the case 15 minutes later, I wouldn't give the lecture because there wasn't quorum enough. So the bacteria do the same thing. Only if there are enough individuals, they will do, for example, formation of a biofilm or promoting an infection. So it's very important for them to be able to talk to each other and to know how many of them are present. And so they use something that is called quorum sensing mechanism, by which the bacteria release certain molecules that can recognize certain proteins at the membrane of other bacteria. And so in this cycle, where actually by this recognition process, they actually change certain genes, and this is why they do certain actions. So sometimes they produce molecules that are relatively easy to see in surface-enhanced Raman. And so the idea is that we have our substrates, and we either extract liquid from a culture of bacteria, or we actually, and this was the main objective, we try to see how bacteria grow on a surface by simply shining a laser and reading the signal from this molecule here. And so with these substrates that I just showed you, it was actually clear from the beginning that we were able to see extremely low concentrations of this molecule, even down to below nanomolar, which is more than enough to actually see only a few bacteria floating around. And so we can actually even find a calibration curve, so we can do this almost quantitative. And then we incubate bacteria in the presence of these substrates. The bacteria, as you can see here, are quite happy on top of the pillars that we have. They do their thing. We shine a laser, and with time we see an increase in all these peaks that you see here, and then we know that the concentration of piocyanin is gradually increasing, and then we can correlate this concentration to the behavior of the bacteria. And so, in principle, if we obtain subsequent funding, we should be able to take this, implant these materials, and actually, for example, monitor what is happening inside of a body after surgery, and to see whether infection is likely to occur or not, so that actions can be taken against that. Okay, so with this, I would like to conclude. As I said, the main message is that there is no golden color, but secondary messages is that by combination of colloid science and nanoplasmonics, we can obtain new materials, new properties, new applications. We can actually tune the colors as much as we like, and we can find applications in very important processes. We looked at biosensing, but other people are looking at energy, at environmental research, and a few other things by using similar properties. Thank you very much. Thank you for the award, and thank you for your patience.